Thank you, Dr. Bienenstock, for this excellent talk. I'm sure there will be a very interesting discussions afterwards. And now it's my pleasure to invite Dr. Karen Ann McVey Neufeld to give her talk. Dr. Neufeld is a senior postdoctoral fellow at the Brain Body Institute of McMaster University, and she obtained her PhD from McMaster with a work on the behavioral changes in mice raised in the absence of microbiota. She was the first to highlight the importance of the microbiota in normal brain function. She continued as a postdoctoral fellow at McMaster and then at University College York in Ireland. And she's particularly interested in animal models of early life adversity and stress that occur during critical windows of development, such as early postnatal life and adolescence. She holds a joint fellowship from the CHR and the Canadian Association of Gastroenterology. So please, Dr. McVey Neufeld.
Hello, I'm Karen Ann. There's your thank yous. So I'm a senior fellow working with Dr. Bienenstock at the Brain Body Institute at McMaster, and I'm a neuroscientist, and I work in preclinical, so rodent models of the comorbid psychiatric and gastrointestinal diseases. So that's sort of where my interests lie. And I use behavioral neuroscience techniques, molecular biology, and some enteric electrophysiology to explore my interests. I'm not going to talk about electrophysiology today, though. I'll stick with behavior and a little bit of molecular. Okay, so I'm going to talk today, again, a continuation of the microbiota-gut-brain axis, but I'm going to highlight a study that I've been doing at the Brain Body Institute that's sort of a continuation of Sophie Leclerc's study that John presented, so the low-dose antibiotic exposure, but this time looking at the critical window of adolescence as opposed to the perinatal period that she published in. Okay, so a lot of this you've now heard, and I'll just very briefly go through and say that I'm also working in the microbiota-gut-brain axis. We know that this is a bidirectional mode of communication between the gut and the brain. And so we have top-down control from the brain to the gut and bottom-up from the gut to the brain. And it's constant, and it's always communicating under normal situations. We know also that many psychiatric and GI disorders are comorbid, and that's where a lot of our interests lie. The microbiome that Dr. Bienenstock introduced is obviously of interest to everyone here, but suffice to say there are an enormous amount of them in us. They make up about 1.5 kilos of our body weight, which sort of leads to the question of who's running the show. Is it us or is it them? Are we just a vase for them, or are they at our beck and call? And it's likely a mutualistic relationship that we have with the bugs. So in preclinical work, there's a number of methods we can use in order to ask some of our questions. One of the things, like I said, as I started with in my PhD work, was looking at germ-free animals, so mice that had been bred and maintained in the total absence of bacteria, so had never been exposed, and looking at how their brains developed and how those animals behaved. There are other experiments you can do, which I'll highlight today, the antibiotic studies, so where you give an antibiotic and disrupt the bugs that are existing in the gut, so you confer some kind of intestinal dysbiosis and then see the results of that. And those studies are interesting because, as John said, you can look at critical windows or different developmental periods and see if it matters when you're introducing that kind of dysbiosis, what your outcome measures will be. You can also add bugs, so probiotic studies where you're adding sort of beneficial bacteria, the good bugs to the system, and see what happens. And there's also the pathogenic study, so adding pathogenic bacteria or an infection and then seeing what the outcome is there. I'm not really going to talk about those types of studies today, nor the fecal transplantation studies, but John's already gone through that for you. So this is a bit of a repeat. Again, this is just highlighting the pseudo paper that John had mentioned. So this was published in 2004, and this was the really landmark paper with germ-free animals that showed that if you looked at a germ-free versus just a normal healthy animal with its intact microbiome, and you subjected it to a 30-minute restraint stress, he saw that the germ-free animal had significantly increased plasma levels of adrenocorticotropin and corticosterone. So just because they had not been exposed to bacteria throughout their life, they had this heightened, what we call HP axis responsivity or stress responsivity that you could measure in the blood plasma. And interestingly, he found that if you introduced bacteria to these animals prior to 6 weeks of age, but not afterwards, that you could normalize this stress responsivity. So indicating that there was likely a critical window, at least as far as brain programming of stress responsivity occurs, that you had to give the bugs sort of early in life, before early adulthood at least. So in 2011, I published a paper that looked at the behavior of these animals, because he had only looked at the stress response and not really went on to see if their behaviors were different. And what we found was that germ-free mice showed a reduced anxiety-like phenotype. So sort of counterintuitive to that hyperstress responsivity, but they showed less anxiety-like behavior than a normal control animal, and that this was associated with changes in brain neurochemistry. And that was quickly followed up by Rochelle Diaz-Heitz. She also showed reduced anxiety-like behavior in the germ-free mice. And then the Cork group also was publishing the serotonin behavioral results. So just very quickly, what I was observing, again, I used the elevated plus mace, which John's shown you, but in this test, this behavioral test, we see that a germ-free animal will spend more time and enter more often into the open arms of the maze, so showing an anxiety or an anxiolytic-like phenotype as compared to a normal animal, and that this was associated, when we looked in the hippocampus with in situ hybridization studies, with an upregulation of brain-derived neurotrophic factors, so BDNF, and that was consistent with the behavior we observed. So no bacteria, decreased anxiety-like behavior, increased brain BDNF. So, I mean, people will argue, and probably rightfully so, that it's hard to make clinical correlates with germ-free animals. So how clinically relevant is a germ-free animal? You're not going to ever see that in a patient population. But this provided, those studies were sort of landmark because they were the first to show that the bugs can actually influence the brain and the way it functions. So that was really important and sort of a, as we say, a landmark paper. But we moved quickly into antibiotic studies, as did a lot of other groups, because you wanted to something a little bit more clinically relevant. If you disrupt these bugs, do you also change CNS function and brain neurochemistry? So most recently, I've been working on a study at McMaster that is a continuation of Sophie Leclerc's study that did the low-dose penicillin that she gave during the perinatal period. And then she looked, so the antibiotic in this case was given to the mothers in her drinking water. And she received that, I think, in the last week of gestation and then up to weaning. And then the pups were grown up into adulthood, and then their behaviors were assessed. And then a number of tissues, including the brain, were examined. But I'm interested more in adolescence as a critical window of neurodevelopment. And this is partly because it's been pretty neglected. I mean, it is probably the second most vulnerable and plastic period for brain development after the perinatal period, and it hasn't really received much attention, particularly with respect to the microbiota-gut-brain axis. And that's unfortunate because we know that teenagers and adolescents are exposed to an awful lot of antibiotics. Particularly, they can be on antibiotics for several years in the treatment of, say, acne, and that's quite frequent in North America. So Adolescence, this is a review that I published with the Cork group last year, 2 years ago now. And in it, we just really wanted to highlight that we all know that adolescence is a period of time when the young adult is exposed to an enormous amount of stressors, be it alcohol, stress, poor nutrition, poor sleep, drugs of abuse, prescription drugs like antibiotics. And this is all happening at a time when the brain is still really developing. And we also know that this is the period when psychiatric disorders often first manifest. So the question is it possible that the gut-brain axis is mediating some of these changes that we're seeing in behavior or the onset of psychiatric diseases? And I like this slide just because it shows sort of everything coming together at once, because you do have this kind of perfect storm where the bugs are developing throughout life. They're becoming more complex in adolescence and as they increase into adulthood. At the same time, you're getting major changes in the brain where it's very plastic. You're getting reduced synapse formation. You're getting increased myelination. And at the same time, like I say, the onset of an awful lot of psychiatric diseases. So this was my study, which is very similar to Sophie's. So I had the same 3 treatment groups. So the water control group delivered to both male and female bulb C mice, or they were given into the penicillin V treated group, so the antibiotic treated group, or they received the co-treatment of antibiotic and lactobacillus JB1. So the 12 hours on, 12 hours off. But this time, they were given these treatments at weaning. So they had been left with their mothers for the normal nursing period postnatally, but from 3 to 6 weeks, they received their treatments. And then I waited 2 weeks, and then the behavioral tests started, and I did 1 behavior per week just to break them up a little bit, not too stressful. And then at the end of the study, they were sacrificed and the tissue was collected. And throughout, we collected fecal pellets in order to analyze the microbiome, which is still in the male. Okay, I just wanted to quickly show some of these behavioral tests because I didn't know how familiar some of you were with rodent behavioral tests, but this is just an open field, and it's very simple. It's just a box, basically. So you put your animal into the center of this field, and then you leave it for 30 minutes, and you leave the room, and you allow it to just explore. And you can measure just general locomotor activity, or you can go more specific and start to look at things like time spent in the periphery versus the center, number of entries into the center, because a more anxious mouse will spend more time in the periphery of the field, and a less anxious mouse will start to enter into the center. And you get these beam breaks that will give you all your data at the end. You can also measure things like rearing, so it's like a risk assessment where they go up on 2 legs and they're sort of looking around. And we conduct this test in the evenings during the dark cycle, so when these animals are at their most active. Okay, so all of my graphs, I think, are set up so that the males are on the left and the females are on the right. The black is the water-treated control, the green is the antibiotic-treated, and the blue is antibiotic JB1 co-treated. So for the males, didn't see too much in terms of center entries in the open field, but we did see a reduction in rearing behavior. So that reduction in risk assessment was observable in the males after they had been treated in adolescence with antibiotic. We did not see that decrease in rearing if they were co-treated with the probiotic JB1 and antibiotic. Females, however, showed a reduction in the number of center entries in the open field after antibiotic administration. So they showed an increase in anxiety-like behavior this time. So actually opposite to what Sophie observed, where she saw the anxiolytic-like behavior, I'm seeing anxiogenic-like behavior when you change that critical window that the antibiotic is being delivered. And that did not seem to change with the JB1 co-treatment. So the females in this case in the open field are showing increased anxiety-like behavior that was not ameliorated with concurrent JB1 treatment. I then used the next week later the light-dark test. So this is another measure of anxiety-like behavior in rodents. And in this case, you put this black box with a little door in it, I think you can see, right into the open field. And you allow the mouse again to explore, and a more anxious mouse or more anxiety-like behavior, they'll spend more time in the dark portion of the light-dark box and less time out in the light, and vice versa. And then you can again measure number of entries, time spent, all those types of things. So this time what we see is the behavior in the males, and this is sort of interesting just from a behavioral neuroscience point of view, is that while we were testing both anxiety-like behavior in the open field and the light-dark, you start to see differences in your results, and that would maybe indicate that it's important to try different numbers of tests and behavior to see what your phenotype is. Because we did see the same type of behavior. So again, an anxiogenic-like behavior increased anxiety in the males this time, but in the light-dark test. So they showed a reduced number of entries into the light, or sorry, this is time in the light, but it was also reduced entries. And again, a reduction in rearing after they had been treated with antibiotics. But the males, this behavior was ameliorated with JB1 co-treatment. So unlike the females, they seem to be responsive to the probiotic treatment that was delivered concurrently. The females, we didn't see the anxiety-like behavior this time in light-dark. So the same behavior, anxiogenic, in males and females in the different tests, with the males responsive to JB1. So the next test I did the following week was the olfactory discrimination test, and this I took from Jackie Crawley's group at the NIH. So she has developed this test where essentially it's really simple. You deliver an odor at the end of a Q-tip or a cotton bud into a cage of a singly housed animal. And you measure the amount of time that the animal spends sniffing the bud. So that's the amount of time where they're interested in interacting with the odor. And you present the odors 3 times, so you should see a habituation in their interest. So they start to spend less time, if you're presenting the same odor over and over, less interest in that particular odor. But if you present a new odor, they should show a renewed interest, and you see that with increased time spent sniffing again. So you'll see it in the graphs when I show you. But you can test both discrimination of non-social and social odors. So our non-social odors are bananas and almond essence, and our social odors are, we basically take one of the Q-tips and drag it through the dirty bedding of an age-matched, sex-matched, strain-matched animal so that they'll know they smell a different animal, basically. And you can present cage 1 and then cage 2 and see if they're able to differentiate between 2 different social odors. And in a control situation, like a normal situation, they should know the difference. So the males are on the left, females are on the right. Controls are on the top, then the antibiotic treated, and then the antibiotic JB1 co-treated. So you can see at the top the way they're supposed to behave. I hope these graphs came out okay. You see a habituation. So in this case, we presented water, then almond essence, then banana essence, and then 1 dirty cage, 2 dirty cage. And you present each of these 3 times. So you see, under a normal situation, a habituation. So they show less interest after that repeated exposure to 1 scent. Then you change it to, say, banana, and their increased interest by increased sniffing time, that again reduces. Almond, same thing. Renewed interest begins to decrease. The huge spikes that you see on all 6 of these graphs are the first time we present a dirty cage. So they're clearly more interested when they smell another animal. And all groups, regardless of the treatment, were interested in a social odor. And they began to habituate, all of them. Pretty much across the group, the female antibiotic treated a little less habituation perhaps, but still habituating compared to the first time. What gets interesting is in the middle 2 panels are our antibiotic treated. When we presented that new social odor, as you can see at the top, you get this new spike of interest. So they recognize this is a different guy I'm smelling now. And the males clearly, and the females too, it's less, but it is significantly different, they can recognize that this is a new cage. Whereas in the antibiotic treated, they don't seem to recognize that you're presenting a new social odor. So they're showing deficits in social odor recognition. Both males and females, this ability to differentiate the 2 social odors was restored after JB1 treatment. So we were very interested and excited about this because, as you can imagine, with rodents particularly, olfactory discrimination is behind a lot of their social interactions, how they process that information, how they would communicate with one another. The last thing that I did was to look at stress reactivity, similar to what I showed you with pseudo, males on the left, females on the right. And there are differences here in that everyone responded to the 30-minute restraint stress as you would expect, the increased blood corticosterone plasma corticosterone levels, so increased stress response. With the males, the antibiotic didn't seem to heighten that increase in stress responsivity anymore, but we did see a reduction in corticosterone after JB1 treatment. This is consistent with experiments we've done time and time again. We see JB1 reduces that stress reactive hormone if you feed it. And we've actually just published in Frontiers in Neuroscience a new paper showing, again, bulb C mice have a reduction in stress reactivity after feeding with JB1. Females, however, they show a significant increase in stress responsivity after antibiotic treatment compared to the control water-fed animals. And again, they weren't responsive to the JB1 treatment in the same way. So they still showed a heightened stress reactivity whether they had JB1 or not. So that's interesting because if you remember from the anxiety-like behavior, it was the females that didn't show an amelioration or an attenuation of their anxiety-like behavior either with JB1. Now this data is actually like from yesterday, so it's hot off the presses. We're beginning to look at the brains of these animals because these experiments were conducted a while ago. And we're now looking at the brain tissue, and we're looking at mRNA gene expression in various brain regions. And the first thing we decided to pursue was these differences that we saw in social recognition. So I decided to look particularly at the hypothalamus, which we know is important, and there are certain genes that would be targets for deficits in social order recognition, and particularly, in this case, I'm showing the oxytocin receptor. So what we saw was a significant upregulation of mRNA expression of the oxytocin receptor in the hypothalamus of these mice. Now, I've coupled the males and the females together here because they actually both showed the same thing. So the data is combined. They both showed an increase in oxytocin receptor expression in the hypothalamus that with JB1 treatment was reduced, or normalized, I should say. So this is interesting because it could potentially point to a mechanism or something that we need to look at more with regards to the deficits in social odor discrimination. So we're going to continue this looking at AVP and AVPR1A, so the arginine vasopressin proteins receptors 1A are particularly important for social communication. I've already looked at the olfactory bulbs and not really seen too much difference in gene expression there, but so right now we're going to target the hypothalamus, and then we're going to go on to the hippocampus and frontal cortex. But we were very excited, or I was extremely excited about this yesterday. So in summary, for this study, both male and female bulb C mice show long-term behavioral changes after adolescent treatment with low-dose penicillin V. So similar to Sophie's findings, there are long-term problems that we're seeing as a result of these treatments, but this is a different developmental window. Both males and females showed the increased anxiety-like behavior, albeit in different behavioral tests. So we saw the open field and the light-dark test. Both the males and females showed the deficits in social odor discrimination, normal for non-social odors, but definitely couldn't recognize the second cage when it was presented, the odor of the second cage. And this was associated with changes in the hypothalamic oxytocin receptor mRNA expression, and these changes were restored following the concurrent probiotic JB1. And the females showed the increased stress reactivity following the adolescent penicillin exposure, but that didn't seem to be attenuated with the concurrent JB1. So the implications are probably fairly obvious. We need to consider the potential negative long-term effects of antibiotic exposure in adolescence. It's something that isn't really, well, perhaps now we're paying more attention to antibiotic prescription, but we haven't really explored, I think, that there could be long-term CNS changes or something occurring at the level of the CNS after these antibiotics, and that the males and females may not be responding similarly, and the importance of looking preclinically at males and females, but also clinically. It's exciting that there are partial preventative effects of the lactobacillus rhamnosus JB1, and it's encouraging in terms of looking at potential treatments in the future for these kinds of antibiotic exposures. Where the study is going now, I'm particularly interested, as Elena said, in early life adversity, and I've done a lot of work in doing maternal separation in rats particularly, but lately I've been developing in our group a mouse model of early life adversity where the mothers are presented with very little bedding, so it's called a limited nesting model of early life adversity where the moms just aren't given the bedding to make a nest for the pups, and they end up neglecting the pups quite significantly. So it's a model for significant neglect and early life challenge. And I'm interested in coupling this because we know that that's a risk factor for going on to develop psychiatric and gastrointestinal diseases, certain gastrointestinal diseases. But not everybody goes on to develop these things. So there's this idea that there maybe a double hit is necessary. So you need to have a second challenge at another vulnerable period in life. So that would be, for instance, when the antibiotic is introduced in adolescence. So if you have a background of early life adversity coupled with that second challenge, what then happens? And that may be helping us to understand some of the etiology of these comorbid psychiatric and GI disorders. So that's that. And then thanks to our students. So Sebastian Kay is our undergraduate and Kevin Champagne-Jorgensen is our PhD candidate. And they both helped out a lot with the molecular work that I showed today. And then John Bienenstock, the director of the Brain Body Institute and my mentor. And thanks to my two funding agencies, CIHR and CAG. Thank you to the Lawsons for providing the opportunity to present my work today and for the invitation, Helena. And thank you all.
Thank Thank you for sharing this novel data with us. It was very intriguing. I think I'm going to invite you to please sit at the table for our question and answer session. So for anyone who has a question, we have a microphone and Lorena will help. Bring it to you. Yeah.
Very interesting presentation and very interesting talk. We are sort of full bacteria in everybody, and there are so many different species bacteria. There's so numerous bacteria that is so-called microbiota. So when you study this microbiota in your experiment, I know that there's numerous. I mean, what do you think about any effect of some specific species of bacteria or a combination must be there to have some effect?
Are you saying why? Sorry?
Yeah.
Why is the JB1?
Yeah, I mean,
How do we select?
Yeah, well, you in your study you present a lot of data about microbiota change, and for example, you treat penicillin. Certainly, you will reduce many different species of bacteria. I wonder what bacteria really matter. You say it's a general fact, or maybe it's because certain species or certain specific bacteria, their balance is increasing or decreasing is really important, or just the balance of different ratio of different bacterial species or specific bacteria could be makes a difference.
I think you're striking. I think you're hitting at the core problem that we have. We use one bacterium, basically not because we think that one bacterium can do everything, but we because it's easier to try and then dissect out what the pathways are that are basically involved. In the clinical situations now, there is a big growth in the development not of probiotics, single probiotics, but in the assembly of communities of bacteria, which may be able to give produce multiple effects. And this is only just beginning. So the question of sort of if you're hoping, or as we hoped originally, that we'd find the bacterium, and people are looking for the bacterium that's missing, that's not been proven to be very helpful at the present. So I don't think that we think that that this particular organism that we use is the only organism. But interestingly enough, when you carry out studies, as we have done, with, say, a single organism, let's say it's JB1, and then we see certain things that we want to reproduce, we carry out the same set of experiments with another organism, which is also positively ineffective. When we put those two organisms together and feed them, basically, we often find not synergistic effect. We find counter effects which actually wipe out the effects. And the problem with all of this is we don't know what's going on in the intestine at a particular site as a consequence. So I'm just, if you like, introducing to you the fact that we know very little and really can't offer the best advice in terms of which organisms are going to be successful.
Hi, thank you for the great talk, John and Carrie-Ann. So I just have 2 quick questions. So first question was, so you've shown the data for both experiment with the adolescents and also the early life feeding of the JB1. And I was wondering, did you guys ever did, just a JB1 control in non-antibiotic treated mice and seeing whether it had any effects? Because it certainly looked from the adolescent data you just shown us that, yes, the antibiotic JB1 reversed the effect of the antibiotics, but it's actually slightly lower on the elevated plasmase test than the actual control animals. So I was wondering whether it had any effect on its own.
Yeah, we've done a number of tests now, Richard, looking at JB1 treatment in just a normal control type situation. And actually, we've got a paper coming out this week, I think, in Frontiers, just showing the effects of JB1 treatment both in the tail suspension test, which is a measure of depressive-like behaviors in rodent world. So how it's sort of, it's a measure of depressive-like behaviors. I'll leave it there. So what we see is if you give JB1 treatment, you see a reduction in those depressive-like behaviors just in the normal situation. And again, same with the stress responsivity. You see an attenuation in the HP axis. But we also have a lot of other behavioral tests data in the EPM and that sort of thing as well. Interestingly, though, it seems to be strain-specific, which is actually the point of our paper, that we see it in bulb C mice, but we don't see it in Swiss Webster. And that's because we think that you need to have that genetic vulnerability or predisposition to anxiety-like behavior in order to see the effects of JB1 similar to, say, an antidepressant. You need to have that genetic vulnerability first, or else if you give, an antidepressant to a normal healthy animal or a normal healthy human, what are you going to measure? So that's what we're sort of focusing on there.
Another question is...
Just to add one other thing, it's interesting because we're also looking at the moment at antidepressants and how they're working, and are they working potentially through the bacteria and so on and so on. So that's another end of it. But Prozac, for example, which was one of the first SSRIs that work in clinical depression and anxiety, basically is well described as not working in normal human health. You have to have the issue that you have to have the anxiety, you have to have the depression, and then you can see that it's working. So that's the counterpart to what basically what Carrie-Ann was saying.
Mm-hmm. And just a follow-up, so in the adolescent experiment, because for Sophie experiment, they show that microbiome changes are quite resistant to the JP1 exposure. I was wondering, did you measure, microbiome for your adolescent data, or are you smiling already?
Yes, I'm waiting to hear back from the microbiome analysis. So it's at the institute right now getting analyzed. So we've collected it throughout life, and then we'll see.
Thanks. So I think my question might be related to the last one, actually, which is sort of zooming out, thinking of humans. The lactobacillus bifida specific to each species are really key here. And so it really is important to tease out what they do in the presence of antibiotics, right, relative to everything else. And so is there more information on how lactobacillus presence affects obesity, behavioral outcomes in the various mouse models? Because for humans, the epidemiologists are now trying to track early antibiotic exposure as a risk factor from childhood overweight, later obesity, depression. So they're looking at that cross-sectionally and with cohort studies. But I'm guessing in the mouse model world, there's been work to kind of tease out some of those life course associations. Could you tell us a little bit more about those, please?
The reason that we did the low-dose penicillin is work done by Laura Cox, basically, who described it in a maternal obesity model. It wasn't looking at behavior, and showed in a particular strain that similar treatment with low-dose penicillin actually promoted obesity, but only when challenged with, it didn't promote obesity on a normal diet, it promoted obesity when challenged with a high-fat diet. Now, and that was what triggered the experiments that Sophie Leclerc basically did and that I described. But the additional reason that we did this was because in the literature, people have looked at trying not single antibiotics, but multiple antibiotics, and overdosing, in essence, with multiple antibiotics. So the problem, if you look at the literature, is you see the results after antibiotic treatment, massive overdoses, basically, when it really comes down to it. They're non-absorbable, basically, mostly. But the constellation of antibiotics that are used by people in the literature is really horrendous. So we're just publishing a whole treatise, if you like, and review, pointing out that unfortunately almost every antibiotic that's been tested carefully at some dose, basically, has an effect on the enteric nervous system, which is quite recordable, and or the brain. And there are, in fact, clinical syndromes in terms of even penicillin to people who are susceptible to induce epileptic attacks. So it's very complicated, and we can't exclude basically the fact that the antibiotic effects that we're seeing in part, everybody assumes it's because of the bugs. But we can't make that assumption, and it's potentially dangerous, but very difficult to resolve what it really is. It remains for many people to repeat the experiments that we've done, that Karen Ann is doing, and also to try other things. So the answer, I'm afraid, is very complicated, but it isn't necessarily only the microbiome. The bacterial microbiome. What I haven't talked about, of course, is the viruses, as an example. 60% of the genome, basically, in the gut that is microbial is in fact phage, and then now we're into another world of what the viruses are doing, and that's why basically I think it's interesting that the maternal immune activation models, which are poly-IC, which mimic a viral infection, are in fact possibly much more important than any of us have thought so far, because almost the whole of the literature at the moment is on bacteria.
Thank you for your great talk. I just had a really quick question regarding your data point using JB1 treatment and the dampened corticosterone response that you observed following, I think it was a social defeat model. So I'm not really too sure on the methodology of providing the treatment, whether it was oral gavage or it was free drinking. Was it oral gavage?
The experiment that I showed was delivered via the drinking water, but we've also done it with gavage. We've seen it in both. Yeah, because I tend to try to avoid the gavage because it induces, it's quite stressful.
Exactly,
if that's
right.
what you're, yeah.
Right, so that was the exact question I was leading to, and I was wondering if there was any control that you had conducted to prevent or to understand the effect of HP axis programming prior to. Okay, so you've done both.
Yeah, see, that's the conundrum, because, I mean, ideally, the gavage is great because you can quantify better how much bacteria you're delivering versus making a kind of a guesstimate when it's in the drinking water bottle. But you know, you're handling your animal daily, you're gavaging it. I mean, we do have the experimental controls where you're not gavaging bacteria, you're gavaging like a PBS or something to the animal. And we still see the reduction in stress responsivity with JB1. But you're right, absolutely. You have to weigh the pros and cons of, do you really want to know exactly how many bugs are getting in there, or do you want to give it in the friendliest manner possible so that the animal is chill?
Okay, great. Thank you.
I'm sitting here feeling the weight of Department of Nutritional Sciences above us, and so I feel compelled to attack a technical question from that perspective. And the question is, if memory serves, vast sums of money and years of effort were put into defining the dietary requirements of mice and other experimental animals under various conditions. And I'm sure you're all using well-controlled, well-defined diets. The question comes to mind after your talks, did anybody ever bother to repeat all that work with germ-free mice? Or are we using the same defined diets that have been defined for normal mice with normal microbiota? And if that's the case, then it raises the question, are you perhaps missing, in the absence of bacteria, some metabolic requirements that in a normal mouse are met through the combination of these defined diets with bacterial action, but in the germ-free experiments, you've lost that. And so now you've got an artifactual situation that's confounding all of your interpretations. Please.
No, that's absolutely true. That's one of the reasons, like I was saying, that you move away from some of the germ-free models because it's rife with problems, and one of them is that they definitely metabolize their food differently than do a normal, because you need your bugs to break down the food properly, and if you're missing those bugs, then the food isn't getting broken down. You're not accessing the same nutrients. So they have a nutrient supplemented diet in order to try and compensate for some of that with germ-free animals, but it is one of the problems that you can't really differentiate. I mean, they're having these metabolic problems due to the absence of bacteria, but are some of the effects that you're seeing due to the fact that there may be nutritionally sort of subpar, or is it, it's an indirect effect of the bugs?
The other thing, I mean, we're not happy with you doing too many germ-free experiments because of that, and in trying to. Yeah. Yeah. No, I mean, I'm happy to agree. Yeah. Well, people have tried. Some careful people are trying. I mean, I think the thing that really drives us these days is how expensive it is. The sort of experiment that I was talking about, for example, the low-dose penicillin, it's multiple. First of all, it's several months of work. So, secondly, it's very expensive, and therefore it is extremely difficult to get this done in academia. When it really comes down to all of the pressures and so on publication and students and so on and so on. I'm not defending it. I'm just saying that I agree with you. I think there are major issues associated with that. I'll just give you an example. We moved our lab 10 years ago, whenever it was, from McMaster to St. Joseph's, basically. I was doing certain immunological experiments at that time. There was using a specific model of colitis at that time, standard model using DSS, dextran sulfate, which gives a chemical colitis, absolutely standard at a certain dose in the drinking water. And basically, so average out there, 2% will do it. It will give a very defined set of a set of conclusions. And we were doing it regularly at McMaster. When I moved our lab to St. Joe's, basically, we couldn't repeat it. And so we tried 3%. Then we tried 4%. 4% is sort of lethal in everybody's hands around the world. We went up to 6%, and we didn't get colitis. So what was going on? Well, we don't know. We have a clean new facility, basically, versus the standard facility at Mac. So those are the sorts of issues that we're all, I mean, any of us who are in the game, actually facing. And it's not exactly what you were asking, but it tells you that the surroundings, everything from the food to the surroundings are crucial.
One thing that we didn't speak about today really or present any data was on prebiotic studies. So in this case, you're really giving the food to the animals that the bacteria use to break down and benefit from. And I know in my previous fellowship, I was at the UCC in Cork, and it's sort of a enormous lab with lots of different postdocs. And all the postdocs would always say that really the prebiotic studies and not the probiotic studies were some of the ones where we saw the most robust findings in behavior. And it was so we were providing that sort of foodstuffs to the bacteria, not really interfering you were interfering with the bacteria indirectly through the food, but you were seeing really major changes that were pretty robust with prebiotic treatments as well.
One or two more short questions. So please, yeah.
Very interesting talk. I have just few very short questions. So the first one is, I think the question have been asked by Richard, and it's related to the microbiota. And then do you think that the effect of the rhamnosus you are using, the GB1, is mediated by the microbial changes in the gut, or it's a direct effect?
I don't know. I don't know.
And the other question, why GB1? Why did you choose GB1 and not another rhamnosus? Is this because of strain specific effect?
Yeah, I can tell you exactly. It's, the honest truth is that we thought it was something else. We bought it from the ATCC, which is a standard American tissue type culture collection. We thought it was a Reuterie. We used it. We lost the effect. We ordered it again from, this is 12 years ago. We ordered it again from the ATCC. We got the bug. It didn't work because we think that they gave us something else, because we then just said, well, what the hell is it? And it was turned out to be a rhamnosus. It wasn't a Reuterie. So basically ATCC say, you messed up. It's your problem. And we have absolutely no idea where it originally came from, except that it works. And we are using the same thing over and over again because we have the seeds and we have it stored. So the short answer is we haven't got a clue what it is. We don't know where it came from. It could have been a dog. It could have been a worm. It is a rhamnosus. Yeah. We have sequenced it. It doesn't help us sort out what it is. Yeah. And that's the reason it's not patented, because you can't patent it because you don't know where it came from.
Yeah. Do you have the genome sequenced? Sorry? Do you have the genome, the entire genome?
Yeah, sure we have the genome. Yeah, of course we do. Yeah.
Hi. So more broadly speaking, is there any interest in identifying fecal biomarkers of dysbiosis in the context of mental health? So I'm thinking of a lower cost way of aiding in diagnosis and treatment as opposed to sequencing every sample. And in the future, how likely do you think it is that that kind of information will make its way into clinical practice?
Are we doing anything in terms of biomarkers?
Or just the field in general? Yeah, is that going to be integrated with mental health?
The field, it's absolutely clear that it's missing, and therefore you can't think, you can only rely on the clinical diagnosis when it comes down to whether the patient is depressed. You take 5 psychiatrists and offer the same patient, and you'll find probably 3 different diagnoses when it comes down to subtype and phenotype. It's absolutely clear that a percentage, probably 30% of, let's say, major depressive disorder that is agreed upon, actually have a clear-cut inflammation according to biomarkers of pro-inflammatory cytokines and CRP. And IL-6 appears, because I talked about IL-6, appears to be rising in terms of a possible biomarker. There is no pattern yet that is either predictive or, in fact, diagnostic. And people are searching like crazy to obtain them. So we don't have them. Many people are wondering, is it possible that we could fingerprint it if indeed we believe, and it turns out that the microbiome really does have the clue And is it possible that that is going to do it? That's extremely difficult because when it really comes down to it, there is a tremendous problem with, you can identify that some things are up or down. For example, Akkermansia, or something which is basically an interesting bug that is associated with, or F. prausnitzii, which is associated again in another situation with anti-inflammatory effects, with T-cell upregulation, with all sorts of interesting things that are basically anti-inflammatory. So far, any attempt to try and use those as single organisms they're not been mostly published, but if you talk around, they basically haven't worked so far. And then the other problem, which is the major problem, is that it's extremely difficult to find a patient at first encounter who is not on something. And basically, obviously, if they're seriously depressed, they're on something immediately these days, basically. And then the question is, so what's the drug doing to the bugs? And it's increasingly evident that many different drugs, metformin is a classic example, cyclophosphamide is another, that appear only to work as a result of their action on bugs, which in turn are changing the drug into something that's effective. So the problem then is, if you've got the psychiatric patient and you do a fecal sample and then you publish, nobody has a clue, if you really get down to critical thinking, whether or not the effect that is observed, this is up, that's down, is actually due to the drugs that they're on, or is actually due to the disease. And that's the crux of the matter. So if we get the biomarkers, basically, or biomarkers, and there's a lot of work going on around the world on that, then it might be possible to take that person basically with a biomarker, at least as a diagnostic approach, and immediately get fecal samples. We've done one study. We're about to submit the paper with Val Taylor, who's a psychiatrist here at Women's College in charge, and at CAMH, who got 16 patients. It took 5 years to get these 16 patients who presented with depression, who didn't have who weren't on drugs, basically a first encounter. And we've just got the sequencing, we've done all the sequencing of the microbiome, basically on those 16 patients. And there were maybe some interesting things there because they were then treated. We have the microbiome on a continuing basis beyond that. And so we know they were on a single drug, all the 16 patients. The bottom line, and I can reveal the bottom line, was that there is a pattern of bugs that seem to be associated with the responders, the people who respond to treatment, okay? There isn't a pattern that emerges that is diagnostic, if you like, of the condition. So that's where we are at the moment. And I can tell you, I'll be surprised where this paper gets published, because 16 patients does not make for very much. And if you think just think about it for a minute. You know, you're a patient, you're depressed, you finally got to the doctor, and basically, and then the doctor says to you, just hang on a minute, we want a fecal sample, right? You know, it's amazing that Val got 16 patients.
Prior to 1975, we did a lot of vagotomies and pyloroplasties. After 1975, it was H2 blockers. Subsequently, it was PPIs. Anything in the literature that gives us some insight into either depression or the microbiome in those different populations? Because they're huge.
There's a huge population has been looked at in terms of epidemiological demographic information in Denmark, where you can follow these people. There's something like 12 to 15,000 they've got who had vagotomy, truncal vagotomy for peptic ulceration, which is what as you say, was available, and that was done recently. They only looked at Parkinson's disease, and having looked at Parkinson's disease, what they've been able to demonstrate, published, is that in vagotomy, in the vagotomized patients, they have a lower incidence of Parkinson's disease than if they than the controls, because the controls, all sorts of interesting, not sham, obviously, but interesting surgical controls. That's another long story about the vagus nerve and whether the prion or whatever it is which is causing Parkinson's disease, and which is clearly found in that the abnormalities are found in the gut first, basically preceding the actual clinical demonstration of Parkinson's. In fact, is going up the vagus nerve. That, I think, is widely agreed, basically. So we tried to get them to in intermittent teleconversations with these people, trying to get them to look at depression and anxiety in that population, because they still have the population, and I haven't been able to persuade them to do it. There is no such extent experimental conclusion in the literature yet. So I'd love to be able to do it. Paul Moietti, who is our big demographic sort of type guy in GI, tells me that he thinks he could probably get 10,000 vagotomized patients who are still alive in this country. Whether 10,000 is enough, I'm not sure, to do a study like this.
Okay, I think this concludes our session today. I would like, please join me thanking our speakers again, and thank you for sharing with us your expertise. Thank you.